All right, good morning, everyone. Thank you for joining us both in person and virtually. It is with great pleasure and honor that we welcome Dr. Peter Forsyth uh, as our distinguished Grand Round speaker for today. Just to give a little bit introduction about Dr. Forsyth, he serves as the chairman of neuro-oncology program at Moffitt Cancer Institute and holds the position of a professor of neurology oncology at University of South Florida. Dr. Forsyth earned his medical degree from McMaster's University in Ontario, followed by a neurology residency at Mayo, and followed by a fellowship at Memorial Sloan. His extensive contributions to the field make him an internationally recognized physician scientist, especially in the area of neuro-oncology. He led many phase 1, phase 2 clinical trials, including developing a novel cell therapy for leptomeningeal disease from melanoma. He's also a very prolific writer and author, publishing in more than 100 articles in esteemed journals. We are very privileged to have him today, and we look forward to learning from his insights. Thank you, Dr. Forsyth. Uh,
Well, thank you for that introduction and the opportunity. It's kind of great to be here. It definitely is weird, in the post-COVID world with things being virtual, which is fine, but it's like this everywhere. It's an honor to come to neurology because I trained as a neurologist and I still do neurology, at least cancer neurology. So it's kind of fun to come and see what's going on. It's such a huge department and you people are so busy. It's amazing. All I want you to do today is learn and ask questions. I'm going to stop talking at 1:45 at the latest. If you asked me 1,000 questions and I only showed you one slide and you learned something, that would be success. So, I think the slides are a way to try to educate you and get you excited. I've seen them, so I don't need to see them, if you know what I mean. So leptomeningeal disease is I guess, a rare tumor where the tumor cells invade the subarachnoid space. They enter through the choroid plexus. And then they seed in the CSF and patients get really sick. On an average, patients live about 3 months. Well, I think that's a challenge. And there's very few people who work in the field and there's very little that's known about it. So the reason to have these planets here is that life is for LMD. We shouldn't assume we know anything, like we know a little bit about Mars, but all these other planets, of which there's probably going to be billions in the universe, right? And it's, without getting into the religious implications, I believe that it's highly likely that hundreds of millions of those are going to have life. We're not going to really recognize life on this planet because it's going to be really weird. It's going to have its own rules. It's going to follow the usual rules of physics and chemistry and biology. But maybe life will thrive at 1,000 degrees Fahrenheit or at 300 Gs, or maybe there won't be carbon and maybe we won't even figure it out. So I think we need an open mind about LMD, particularly when concluding that there's nothing to be done about it. And I guess the point of this is that there isn't a lot that's been done because we don't understand it and we've just sort of given up, Again, it's a rare disease, so it's hard to work on this when you're called to do strokes every 20 minutes, right? Let's see, so this is supposed to move in theory, isn't it? When I push that button. Yeah, my talk only has one slide. Which is, I'm going to click instead.
no, you can use it now.
Those are my, oh, I had to get going, thanks. So I'll tell you a sort of personal story, and that is that I worked on gliomas for a long time and oncolytic viruses and had a big lab. And when I moved to Moffitt, I was having trouble being funded because I was submitting grants about the same old stuff that I was working on. And the head of our research institute did me a favor and he said, look, you got to stop submitting these grants because they're not going to get funded. It's not interesting or novel. And of course, you never want to hear that. So I was really angry with him, but I went home over the weekend and I thought, he's right. Then I went back to clinic and I saw all these patients that have leptomeningeal disease, and I literally was saying to myself, which is not an entirely, gentle thing to say, but why are these people here? There's nothing I can do to help them. So I was like, well, wait a minute, what an opportunity to help them because they're desperate and nobody knows anything. So maybe we should take a shot at this, try to figure it out. I'm going to kind of summarize things because I want you to be educated about it, as well as go into some of our research and the clinical trial that's based on some of the research we did, which is really just the beginning of cell therapies. Now, oh, geez, we've known about this disease for a long time. Yeah, 1869. That's probably, what is that, 19 years after SUNY was founded, or no, 9 years after SUNY was founded? When was SUNY founded? I think it was the 1860s. In any event, and then it was really the fantastic and amazing Dr. Posner at Sloan Kettering. I'm sorry that he's retired because none of you in your rotations get to meet him. He was an extraordinary neurologist. He loved neurology, so passionate about it. And Dr. DeAngelis began to take this problem seriously and published the first paper in 1974 in the modern era. And then I think one of the first clinical trials to use immunotherapy was the pembrolizumab study. So we're just moving on to make more progress, we hope. And then, what am I going to talk about? What's LMD? A little bit about the biology. How are you going to recognize it? And then talk about our cell therapy trial just started. And then, the goals are, has anyone seen a patient with leptomeningeal disease? It's so bad. But our goals are, we need to keep focused on the goal. We need to prevent it. So we should find ways to stop it from happening. And we should find ways to detect it so we can treat it early. And we should cure it in a way so that people aren't messed up, so they're thinking normally and their brains aren't suffering from the side effects of radiation or all these immunotherapies. This has recently happened with melanoma brain mets, in which I also did a trial that really, when I was a resident, having a diagnosis of a melanoma brain met was fatal. It was one of those things where you didn't want to walk into the room. And of course, now with the checkpoint inhibitors, Ipi and Nivo in particular, at least half those patients are probably cured, actually. Is not a word we've used before. And not only are many of them cured, but they thrive. Once they get over any side effects they have, they're normal. So normal that they have the luxury of developing other diseases. So out of the, say, 13 long-term survivors, I've had 4 have developed other cancers. Somebody else has developed heart disease, and so they're actually dying from other things. So we don't know anything about the epidemiology, but I made it up. So because it's not a recordable disease like breast cancer, etc. So we just do a little hospital series, and we think it's becoming more common. We think it's about 5% of patients with cancer. So that would be about 100,000 people a year. The most common cancers develop this more commonly: melanoma, breast, and lung. We have about 104 patients a year at Moffitt, and then we do have a intrathecal chemo clinic, which is about 15 patients. This is probably a third of what Sloan Kettering does, by the way, which is so big. You probably haven't heard of Moffitt, but it's the third biggest cancer center. It's pretty busy. It's like 150,000 new patients a year or something. So what is leptomeningeal disease? Well, we're doctors, right? So it's not a disease. It's like a person. It's about a person. So this is Sarah. She was about 35 when I met her. Single mother. She had melanoma and got a bunch of headaches. And her cytology was negative, so we had to go on the basis of an MRI scan to diagnose it, which is an opinion, which means we can make mistakes. And the first time she met me, she hated me because, of course, I told her she had this terrible disease. And she said, well, how do you know I have it? Because the tests are all normal. And I'm like, well, on the basis of your MRI, I think that's the truth. I said, well, what do you mean you think it's the truth? This is my life we're talking about. So I'm telling you that story just to say that it's still based on opinion because cytology is only positive about half the time. So you really need a really good neurologist. But in any event, a neuroradiologist. In any event, she donated her brain at an autopsy, and you can see all the black stuff in her sulci and gyri and pia. This is her spinal cord down in the cauda equina, and it's just disgusting. So we used immunotherapy for her, and she got about 3.5 years. During most of that time, she was pretty well, actually. We used this for some experiments, actually, so it really helped. So when I think about people we have to do better for, I think about Sarah, as well as all the young breast cancer patients we're seeing. In any event, the diagnosis of LMD is, sort of opinion, as I mentioned. So there still isn't a really good test. So cytology is positive only about half the time. And then there are companies that have came and went. They're doing like circulating CTCs or other technologies, cell-free DNA. It's still not accepted as a diagnostic test. Sloan Kettering is doing their CTCs and sequencing on the tumor cells that float around in the spinal fluid. That hasn't really been published yet. It's just an example of staining the, what is that? Mauve is the nucleus of the cell, and then the green is for HER2. So if you have cells that have a nucleus and they express HER2, you conclude that they're circulating tumor cells in the spinal fluid, but it's still not an accepted test. And you really want to know about the sequencing issues because many of these, it's not like stroke in the old days. There's a whole bunch of mutations which are targetable in cancer that you really want to know about that can make a huge difference. This is my one of my favorite slides. Again, it's hard to diagnose LMD, but you can see it's kind of going into the sulci. I think they have early hydro. Their ventricles are a bit big. Can't see it that well, but there's enhancement in the IAC down there. And then there's areas in the folia, and they're a little nodular. Oh, and there's something in Meckel's cave that I don't think I'm showing you here. Maybe it's on that side there. So you need a really, really good neuroradiologist. We get misled in the spine. So this is normal enhancement. When I was a resident, the MRIs weren't this good, but now you can see just the superficial veins and arteries on the surface of the cord show you some contrast enhancement, so you can't be misled. What you'd like to be sure before you told someone they had LMD is that it's kind of lumpy, and all this enhancement down in the cauda equina. And of course, this is just an accumulation of tumor cells down here, and then it's just completely full. Dr. Bohr at Sloan Kettering is, I think, the world's leader in the biology. So she published a few papers that are pretty important. You'll hear about some of those. But everybody talks about the blood-brain barrier, which matters to you. Doesn't matter to me. We talk about the blood-tumor barrier, GBMs or brain mets. But this is a blood CSF barrier. So what this is supposed to be is a tumor vessel or a blood vessel with the epithelium of the choroid plexus, where choroid plexus will open and admit tumor cells into the spinal fluid, and then they'll float around and cause all their problems. Through a Cell paper, which is probably the first science paper on LMD, she showed that these cancer cells make a complement that interact with the receptor C3a on the surface of these epithelial cells in the choroid and allow tumor cells to pass from the blood through the choroid and then float around and start drifting everywhere. I'm really not here to promote Dr. Bohr, but it's just she's the only one who's done stuff. So I think the title says it all, that the CSF is a little nutrient poor. It doesn't have a lot of nutrients. There's not very much oxygen. It's particularly low in iron, for example. So it promotes the low levels of iron, promote the tumor cells to express this protein that goes on to support leptomeningeal disease growth, tumor cell growth. And seeing that the CSF was already depleted, she's currently doing a study of this iron depleter to see if you can reduce the iron in the CSF further to make it very lethal to tumor cells. And then some of the work we've done, we have not. I mean, I'm a middle author on this paper, but we have not done a lot of work on this. But there are also vulnerabilities in terms of an unfolded protein response, which you probably know about from stroke and other things. So when a cell's in trouble, it activates a bunch of kinases and signaling pathways, some of which there's inhibitors for now in the clinic. So knowing that these cells are under stress suggests different therapies that we just haven't had time to look at. Just kind of just overall, sort of what we're doing. We'll probably talk about this. So we do single cell, which 6 or 7 years ago was innovative, a little less so now. It's a major tool. And then I'll talk about this. Why haven't we made progress in this disease? One of the problems is that you can't grow cells. So in the cancer world, if you can grow cells and stick them in a dish, you can, oh my God, you can squirt a bunch of drugs on them and see what kills them. So you can do these high throughput screens, test them in mice, and then do a trial. But nobody's been able to grow these until, I think 2 years ago, we were the first one that grew them. Unfortunately, we grew them from an autopsy, from actually Sarah and several others. So we were able to collect a whole bunch of cells at autopsy and grow them in a dish, and now we're doing high throughput screens. Another group in England has done this. And then the other thing that we do is to profile people when we're giving them a therapy. We look at all their cells and their chemokines and cytokines before treatment, sort of on, and then when and if they recur. Anyway, that's sort of. Yeah, and this is really the point of this map is that we're trying to get a map to see where we are and where we're going, to see what we can target. So remember Sarah, right, who I've talked to several times. So this is phosphoproteomics, which is basically, well, it's run on an LCS machine with mass spec, and you get a whole bunch of proteins that are phosphorylated, and then you just plot them all, not knowing what they all are. But this is Sarah now. So these are a whole bunch of phosphorylated proteins, and I don't even know what they all are, but you can see that her lane here is different than the other people that lived a short period of time. She got all this red stuff up here, whereas everybody else who died quickly was all blue stuff the first time so there's something different about her, something different about her spinal fluid that I guess was helping to fight the cancer cells. We have some hypotheses, but that's another story. So I'll show you a bunch of these pictures for single cell when we just profile people with their CSF. These are melanoma patients, which doesn't matter, but these are all the tumor cells in the CSF or in brain tumors and compared to their skin cancers. And their profiles are different. Again, this is like cancer. You just sort of look at these tartans, and you make certain conclusions because these look different than these. And the brain ones kind of look similar to the skin. So there's something new and different that we don't really know in the CSF what those are. And there's more, these are all the myeloid cells. So there's like lymphoid cells, like T cells, B cells, and there's myeloid cells like macrophages, dendritic cells, monocytes, MDSCs, whatever all those are without explaining them. And there's just a greater population of some of these cells in the CSF. So we need to figure out why that's true. It's like technology. I think this is repeating the same thing, so I'm not going to go over it. Even though I put this here. Yeah, so we're continuing to do work on the characterization of the immune microenvironment. So I'm going to skip over this. And again, just trying to discover drugs to treat people. This is kind of what you do. Get tumor cells, grow them in a dish, put them in a mouse. And literally, in terms of drug discovery, you start off with a high throughput screen with a whole bunch of drugs, and then whittle them down to what you think the best drug might be. So we do this with a FDA approved drug library. So there's like 5,000 drugs. Just squirt them on and see what happens, and then see if they have the proper concentration, to see if you can kill them, to see if they're doing what you think they can do, to make sure they have this proper chemical characteristics that you want for a drug to put in a liquid. and then do PK and PD, and then look for toxicity and then do a trial. So it's kind of summarizing drug discovery in 5 minutes or less. I'm not going to talk about this anymore. So, what about the standard treatment? So we do an Ommaya reservoir. That's so you can collect fluid and administer drugs, which can be cell therapies, by the way. And then now we're suggesting that people do proton craniospinal radiation. So that's radiating the whole neuroaxis. But protons are really focused, so they deliver energy in a very pinpoint kind of way. So you can focus on the spinal cord, which you all know has the diameter of about a pen, about the side of a pen. So you can deliver energy to kill the tumor cells in that area without all the toxicity. Using photons, you kind of blast in through the esophagus, GI tract, the mouth. So you get all these terrible side effects, bone marrow, it affects bone marrow and the vertebrae. So this is a way to treat the craniospinal axis without many side effects. We give intrathecal chemotherapy such as Herceptin, that's against HER2, or rituximab against CD19 or CD20 for lymphomas. Try to control the systemic disease, so you need to have a partner with a medical oncology. This is an example of something that made it into Nature Medicine, actually, because it made a little difference. Patients survive from 3 months on average up to 5 or 6 months. And that's like nothing, but it's something, and it made it into one of the best journals in the world. Something else is these checkpoint inhibitors are given intravenously, and Isabella Glitzen, MD Anderson, gave these antibodies intrathecally through the Ommaya, which is well tolerated, and I think people had an average survival of about 7.5 months. Again, this is the teaching part. This is just the proton cranial spinal. Now, some of our therapies have complications, but just to name one, this is the leukoencephalopathy. So people that get methotrexate and were radiated, the radiation interferes with the arachnoid granulation, so it prolongs the half-life of methotrexate, so it can last several days as opposed to several hours, which in turn poisons the white matter. And this is an example of someone who had mantle cell, and I think she's cured of her mantle cell, which doesn't happen very often. But now she has this leukoencephalopathy that developed while she was getting intrathecal methotrexate. The doses of Herceptin, which is an anti-HER2 antibody, which is present in breast cancer, we just need to do phase 1 trials to make sure we're giving the right dose. So again, too much information. Until this study was released in 2018, I've been giving intrathecal Herceptin at the wrong dose for several years because nobody did a phase 1 study. Phase 1 study is where you do dose escalation and then you interrogate the CSF to see what the actual level of the antibodies is. It hasn't been done not because we're lazy or because we're doing other things. It's just hard to fund these things. And I think Emily Leroux was able to do this because she lives in France and probably has more support. These are some of the important therapies. So again, this proton cranial spinal radiation. There's a way of delivering radiation through radioactive particles. So you can deliver them directly intrathecally, and that's Dr. Brenner's idea. So there's now a phase 1, there's now a trial of that. So as opposed to getting radiation to your brain and spine, you're kind of delivering it locally all up and down your brain and spine. I told you about the iron chelator, and this is the Herceptin. We're doing a study of intrathecal Herceptin and Perjeta. Perjeta is another HER2 inhibitor. So we're combining these two, and that's funded by Genentech. Now, where are all the CAR Ts? There's a CAR T study now ongoing now at City of Hope, which Janet Portnow is doing, and that's been going on for about 5 years because it takes a long time to do these, partly because of COVID, because they're a little bit rare to get tumors, so you need to have people come from across the country. The other interesting things are tumor infiltrating lymphocytes, which I didn't spell out, called TILs. So you take a tumor, like a hunk of melanoma, and you digest it, and you can take out the lymphocytes. Then you expose them to a cocktail to make them angry and make them proliferate, and you inject them back into the tumor, or in our case, intrathecally, to see if it'll help a patient with leptomeningeal disease. So that's another example of a cell-based therapy. This is another CAR T for a kidney tumor, diffuse midline glioma. What is this? Oh yeah, this is the one that I'll talk about. So I think this is just, I don't know, advertising or something. Yeah, so great story. I think I told you everything I know about LMD, but it's super boring because we're really just, most of the articles you read are about all the old treatments. You know, chemo, like I'm looking at a review paper now to a journal. It's like all the old chemos that have been given. It's like, stop, you have to do something else. So I think just like CAR T and lymphoma that's transformed the lymphoma landscape as well as multiple myeloma. Who would have thought multiple myeloma would respond to a CAR T? It's crazy. But you know, the next wave is immunocellular therapies. And of course, there's going to be millions of them, but we haven't quite started yet. The point of this slide is that LMD is actually inflammatory, and it activates these innate immune responses as opposed to adaptive. So the innate immune response is, you know, like you get a sliver in your thumb, you know, you can develop antibodies to the wood, believe it or not. But while you're waiting 4 or 5 days or a week to develop antibodies, you need to like deal with the splinters. So there's macrophages that go there, there's neutrophils, there's NK cells. So these are all like temporizing measures. So they're innate. It's just a general resistance. It's not specific for the particular pathogen. Probably not going to talk about this much more, just to show you these figures, but it looks like there's no adaptive immunity in patients that have LMD, but they are activating some of their innate immunity through their NK cells. It's hard, right? Because I don't think you hear these words very often. But the scheme, so the LMD cells are producing interferon gamma is produced by the T cells and NK cells that makes these dendritic cells mature, and then NK cells, they can destroy tumor cells through an innate mechanism. So they go to the cell, they puncture it, they release enzymes, all sorts of things. So it's not enough, though, because the patients are still dying. So we thought that we should develop a therapy that was adaptive or something that was specific to the tumor, because patients' bodies are making this innate response. So can we add an adaptive arm? I think, again, I'm probably not going to go over this. Wow, that got messed up. That's too bad. What happens if I go back? Wow. All right. Well, it doesn't matter. But sort of our approach is to, oh boy. What it's supposed to say is that we're going to deliver these peptides directly into the CSF of a mouse through this little Ommaya reservoir. And then we want to figure out the mechanism, because we don't completely understand the mechanism, and then figure out the processing and how it's activated in the tumor draining lymph nodes. So we do this with Dr. Zanecki, who's the chair of breast oncology and MD PhD. He was in the Rosenberg lab. So there's a whole bunch of screening things that you can do for peptides. There's algorithms, but he kind of, by brute force, looked at a whole bunch of peptides and only chose those that solicited a immune response by secreting gamma interferon. Probably doesn't matter. So for mice and people, what happens is patients get apheresis. It takes about 8 hours to collect their cells. The monocytes are sort of separated out. They get all the rest of their blood back in. You incubate them with you incubate them with these peptides that we know are immunogenic for a couple of days. You turn them on with this cocktail of cytokines that turn on DCs and T cells. And then you test them to make sure that they're actually working and turning on some immune response in the way that you can measure that in the lab. And then we introduce them intrathecally. So this is the pre-work with a mouse. So again, dendritic cells that are incubated for HER2 and HER3. selected a dose that works. So these are HER2 positive mice that have HER2 leptomeningeal disease that we get by injecting the tumor cells into the mouse, and then we inject the peptides. So mice live a lot longer, and they look like they're healthy. They gain weight. 5 more minutes. And oh yeah, so once we get the right dose, most of the mice are cured. So up to 70 days or 120 days, and their body weight's fine. Triple negative is another. So triple negative means there's no ER, there's no HER2, and there's no PR. So there's a lot of young women get this disease, and it's awful. So mice they live longer, not quite as long as the HER2s. Probably not going to go over this part because it won't matter. It's a CD4 dependent thing because, for example, when we do CD4 blocking antibodies, this is the usual mice. And when we do the blocking antibodies, they swing back to be the same as control. So it completely ablates. So it's a CD4, it's probably an interferon gamma thing. This is more lab stuff for you. The mice these are mice that have no LMD. So they do have some cells. This is just single cell where you can classify all the cells. And then mice without tumors. And here I'll just point out that they have hardly any B cells. There's very few. So there's like 20%. No, wait a minute. 8%, 11, 10% are B cells. And then mice that survive a long time have like almost 40% of their cells are B cells. So we know, or we think that B cells might be important, and we're doing those experiments. This is a correlation. So we need to do blocking antibodies. So let's see. So the typical way, is this worse than an MS talk with the neuroimmunology? Explaining these terms. In any event, so the dendritic cells interact with the CD4, which is the helper cells. They're so-called helper cells because they turn on T cells and let them kill stuff. And they also turn on B cells and help them to kill stuff too. So we knew that it was going to do this, although in our case, it looks like it's all CD4 dependent. And what we're discovering now is that there's this other arm where it's B cell mediated and probably antibody mediated. So I'm not going to go through this. The other cool thing we do is once we've treated mice, it looks like at least half of them are cured. So we think this is super important because what we do is we treat the mice, and then we try to re-inoculate. So we put tumor cells back in the CSF, and they won't grow half the time. This is the part I love about this story. If it's true in people, which we don't know, is that it's okay to go through a bunch of treatment, but you really, really want to have a shot and never having it come back if possible. And nobody's talked about prevention of this before. I don't think that, so this is kind of our overall experimental scheme in the last 3 minutes. That there's interactions with these peptide pulse DC1s with the CD4s. CD4s can activate NK cells for NK killing. Some of the NK killing is mediated by an antibody response that I didn't even talk about after the B cell interacts with the CD4. And then there's probably direct cytotoxicity as well from the CD8 cells. And what we really want to do is make plasma cells that make antibodies for a long-lasting response, and memory cells so that the patients will remember, their immune cell will remember. So we took all this, it only took about 7 years, but there's a trial where patients with either triple negative or HER2 positive leptomeningeal disease, we administer these peptides directly into the dendritic cells directly into the Ommaya. Once a week, we kind of alternate the HER2 exposed versus the HER3. And then we see what happens. We've only treated 3 patients at our first cohort. The FDA wanted us to do a super low cohort, which is reasonable. That shouldn't be there. In any event, we treated 3 patients, too early to tell, but it certainly looks like it's safe. 1 patient who had triple negative developed a progressive disease after we stopped the therapy, like within a couple of weeks. So I don't really think it's because we stopped the therapy. It looks like it didn't help her. And the others we're still waiting to hear. So we're going on to other cohorts. The super annoying slides, and I'm almost done actually, because I said I'd be done in 2 more minutes, but at the end of the trial, we're going to look at all the cytokines and the cells and see if we're doing what we think we're doing and what the mechanism might be. In the meantime, and we have to do that all at once. We have to wait till the trial's done, because if you run it on a machine and then run it 3 years later, you're not going to get the same result. But on a clinically available, you can get CSF cytokines from a clinically available panel. So just for the 2 patients that we've studied so far, they do get like a 2 to 5 times increase in interferon gamma, so it's doing something. And then the IL-6 is super weird. It's like 1,000 times higher. And the IL-2 is also a lot higher, and the TNF-alpha is like 13 times higher. You know, and cytokines are tightly regulated. Many of you who've seen CAR T consults in Sloan Kettering, other than you. So they get very high levels with CAR T of the IL-6. The CAR T, one of the CAR T labs is right beside my lab, and they don't see these things. They don't see them this high. And the patients are pretty well, actually. The point of this is only that we don't know if it's going to help or be harmless or harmful, but at least it's doing something, you know? I'm going to skip through this. So I'm going to just summarize now. And that I think for this part anyway, that we think we're activating an adaptive phenotype, which is certainly true. Whether that's going to be helpful in terms of antibodies, we don't know. So we're doing the blocking antibody studies, and we're going to do the chimeric studies too, whatever that means. And then we just need to wait for the results of our clinical trial to see if it's going to be useful or not. I sort of think, you know, we don't know, but we might have to add other therapies like HER2, intrathecal Herceptin, to this priming mechanism, and there's lab data to suggest why we do that. So I think, starting with Sarah and the head of our research institute, who told me I needed to work on something different. You know, it's been like a 7-year story, and we're just beginning to make some progress. And it's, I don't know, it's super fun. I mean, I believe we can cure these people. I believe we can do something for them. There's a big team involved. This is the head of our cancer center. He's really into immunocellular therapy. These are the drug discovery guys. Cernicki's the one whose idea was to do the vaccines, and these are people that work in my lab. That's kind of my funding. So with that, I'll finish, and I'm 1 minute late. Thanks. I wasn't looking for questions on the chat.
That was monitoring questions in the chat, but floor is open if anyone has any questions.
Yeah.
So first of all, you can only diagnose 60% of patients. And MRI is
Flow, did you say?
Sorry.
Oh, yeah, half.
Cytology
Only half.
only half.
Yep.
And MRIs aren't always conclusive either, right?
Correct.
So is it a clinical diagnosis today?
Yes, sir. Yep,
Okay.
usually.
All right. And what's
Which means you have to be pretty good.
Yeah.
Which also means you make mistakes sometimes, right?
And so I know it's been presented like 100 different ways, but what's like the prototypical presentation for?
Ugh. Jeez, I think headaches, kind of just feeling a bit weird. It's not like the old days where you developed a radiculopathy, you know, or bowel and bladder. One of the alerts for me is if your cauda equina is involved, you get this very typical pain. It's like a pain, excuse me, in your butt. And people use this, they do this with their hands. So when that's going on, I'm like, oh my God. So even if you can't see something, you kind of know where it is. Cranial nerve palsies happen, and you have to be, when you look at the MRIs, you got to look at the IACs and all the cranial nerves where they are. But some other, you know, I wanted to put a slide in here about mistakes I've made with LMD. We don't call them mistakes, of course, but things I've learned. And one of them, which you would know, was somebody had a GBM, and he had all this enhancement, which was in his PIA. And of course, what we didn't know but learned later is that he had a big seizure. So of course it's enhancement from a seizure, which you see very, very commonly. So we sort of thought he had LMD. Just before we put the OMI in, we did another MRI and it was all gone. And of course, he was imaged initially with all the enhancement that we mistakenly attributed to LMD. You know, he had a bunch of seizures. He told us about some of them. So that's one way to get fooled. Or if there's any other infectious agent. Anyway, so that's an example of mistakes. We don't call it, I mean, no harm was done, but it's like we were ready to go. Anyway, anything else? Hi.
It's glad to hear you comment on saying the, being of, you know, the senior member, that the old chemotherapy
Yeah.
really didn't help at all.
Yeah.
And so,
Not so much.
again, in the pediatric populations, with a pediatrologist,
Oh,
that, you know, found that we cause more harm than really benefit them.
yes.
And I'm glad that we are now looking at other options of immunotherapy and Parkinson's control. So I'm saying that it's high time, 40 years that we've spent with the old drugs.
Yeah. Yeah, Michelle, that the CAR-T I was talking about was Michelle Monges at Stanford for the DIPGs, you know, and that's a Nature Medicine paper where it's one of the gangliosides, I think. It's against the ganglioside. Yeah, we're just starting. I mean, it's just beginning. It's kind of like stroke, just with TPA, just when it started, you know, it's just the beginning. Anything else? So, oh, yeah. 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 Yes, yes.
How's that different? Same thing?
They're all different. So the GBM story is not widely studied. We have a whole bunch of those patients. I think they don't do well. The medulloblastoma study is, I don't know anything. I'm not going to talk about pediatric neuro-oncology, particularly with you sitting in the room, but it's super interesting because Michael Taylor published a paper where we all thought these were drop mets from the medullo. But what he did is, I forget why, but he thought that that wasn't true. So he proved that it was by hematogenous spread. So we actually sewed two animals you can sew them together and they share their venous system. And one mouse had medulloblastoma and it went to the leptomeningeal in the normal mouse that happened to be sewn to their bedmate. So I think most of the medullos, they would just be treated in the usual way. And then there's a whole family of primary, so the ones with lymphoma, CNS lymphoma, too much information, but you always have to look at the three compartments, the parenchyma, the uvea, and the CSF. So that responds to, you know, CNS lymphoma drugs. And then there's primary meningeal lymphomas like meningeal, well, there's lymphomatosis as well as melanotosis. I'm not sure if that helped, but yeah, they're different. But we can treat those things. We really just need the appropriate targets. Anyway, you know, everybody's busy. I really appreciate you coming and coming on Zoom. And, you know, neurology is great, right? It's so much fun, but it's pretty busy. Thank you very much. If I can help, let me know, by the way. Fun, huh? No, that's good. Thanks.
Right, thank you so much. Appreciate it.
Do I just call the limo?